जी अस्सलाम वालेकुम। हसन कैसे हो बेटा? हसन, कैन यू हियर मी? Okay, the last day we discussed the levels of protein structure. We discussed primary structure, secondary structure, tertiary and quaternary structure. And today we will discuss these four interactions that are found in tertiary structure and in a quaternary structure, how these different interactions are made. And then we will discuss structure of hemoglobin and then the structure of collagen. So let's see these interactions, hydrophobic interactions first. Hydrophobic interactions. So these interactions are between the non-polar R groups of the different amino acids. These amino acids may be in the same polypeptide in a tertiary structure, or maybe in different polypeptides in a quaternary structure. For example, if this is a folded polypeptide in a tertiary structure, and it has this, this part of it contains an amino acid, which is, N, C, and C. This represents an amino acid. This is the central carbon, C here. It has an R group present here. This has double bonded oxygen, and this obviously has hydrogen here. This has hydrogen here. So this is one amino acid. Okay. If this R group is non-polar, and another amino acid in the polypeptide chain, which has a non-polar R group, that we can write this way. It has an N, C, and C. So this C has double bonded oxygen here, and this has an R group here, and this has hydrogen here, this is hydrogen here. So if these two R groups are non-polar, okay, if this is non-polar means it doesn't have any charge on it, and this is also non-polar. So the forces of attraction in between these two R groups or these amino acids are known as hydrophobic interactions. Okay, so these are the weak bonding in between the non-polar molecules, and these non-polar molecules may be, R groups may be represented as CH3 or CH3. Okay, so this is the methyl group, which is part of an R group of an amino acid. We don't need to know the name of an amino acid. Okay, so this is how maybe another amino acid which has a different non-polar R group here, that will also have this hydrophobic interaction. However, these both have non-polar R groups, and so this would be a hydrophobic interaction, right? Then if this is same polypeptide folded, and it has the two amino acids here, this amino acid has, I can write that here, N, C, and C. The central carbon has this R group here, and similarly, it has an amino acid here, which has N, C, and C here. So this is an R group here. Okay, the rest would obviously be the same, and this will be, the same. So if these two R groups one is an acidic R group and other is a basic R group. We can represent this with, this is a variable hydrocarbon chain, so that we can represent with this box. And here I will write C double bonded O and NOH. And here I'll write NH2. Okay. So this part is a basic R group and this part is an acidic R group. I'll write that here, acidic R group, and this is a basic R group. We know that acids, when dissolved in water, they lose hydrogen ion to the medium. So this loses hydrogen ion to the medium, and itself this oxygen will have, because it loses hydrogen ion, so this will have a negative charge on it, and this hydrogen ion is taken up by this base, and this will have, NH3 plus here. Okay, so there is a positive charge on this amino group, and there is a negative charge on this carboxylic acid group. So bonding formed in between these two is known as ionic bonding. Okay, so this is an ionic bonding. This is an ionic bonding. And here, if you these two amino acids in this fold, here, this hydrogen here will have a partial positive charge on it, and this oxygen will have partial negative charge on it. Similarly, this will have a partial positive charge on it, and this will have a partial negative charge on it. So the weak electrostatic force that develops in between this partially negatively charged oxygen and partially positively charged hydrogen, this weak electrostatic force in between this hydrogen and oxygen is known as hydrogen bonding. So this is hydrogen bonding. Why there is a partial positive charge on hydrogen and why there is a partial negative charge on this oxygen, we'll discuss that later in water. Okay, so this is how the third kind of electrostatic or the bonding or interaction in between the different amino acids in a tertiary structure and in a quaternary structure that take place. This even takes place, this hydrogen bonding even takes place in secondary structure. And then if this polypeptide is further folded, and it has this amino acid N, C, and C here, and this is, this brings another amino acid N, C, and C here, parallel to this. And both these amino acids have sulfahydryl group in their R groups. This is an R group of this amino acid, and this is an R group of this amino acid. Both of them have sulfahydryl group, SH on this and an SH on this. This is known as sulfahydryl group. And such sulfahydryl group is found in an amino acid which is known as cysteine. So cysteine is an only amino acid which has this sulfahydryl group. Okay, so if an amino acid has sulfahydryl group on its R group, and it comes face to face, the R group comes face to face with another R group of another amino acid which has sulfahydryl group, this hydrogen will be removed from both of these sulfahydryl groups. Okay, and a bonding is formed, a covalent bonding is formed in between these two R groups. This covalent bonding is known as disulfide linkages. Disulfide linkage. Disulfide linkage. So if we look at the level of strength, so disulfide linkages are the strongest most. Then there are ionic bonding, and then there are hydrogen bonding, and then there are hydrophobic interactions. So hydrophobic interactions are the weakest, and the disulfide linkages are the strongest interactions found in the protein structure. Now we will discuss the structure of hemoglobin, and then we will discuss the structure of collagen. Let's see. Before we move ahead, let's see this question. This is an important question, and by understanding this question, we will be able to identify, the different levels of protein structure. The protein glutenin gives bread dough its elasticity. The diagram represents a polypeptide of glutenin. So this is a single polypeptide. Okay, so when this is a single polypeptide, it cannot be a quaternary structure. This could be maximum the tertiary structure. What describes the structure of glutenin? A quaternary structure, it could not be a quaternary structure. Why it could not be a quaternary structure? Because this is a single polypeptide. And for quaternary structure, there must be more than one polypeptide. So this could not be a correct choice. B, quaternary structure, this could also not be a quaternary structure. So A and B is not possible in this case. Then we have C and D. So what is correct out of these two? Secondary structure, because the loose spiral is an alpha helix. Okay, so this is the loose spiral, and this is obviously an alpha helix. This could be a secondary structure. Then the D choice is the tertiary structure, because the different regions form a 3D shape. Yes, there are tight coiling at these two regions, and as a consequence of this tight coiling, this part of the polypeptide has been turned into a globular shape. Ends of it are turned into little globular shape, or which is a 3D shape. So when there is a 3D shape present, it is coiling and further folding of a polypeptide. It is a 3D shape, or it is a tertiary structure. So when the tertiary structure is there, obviously we will not talk about the secondary structure. Both cannot be true. So the final tertiary structure is there. That's why D should be the correct choice. Okay. Let's see then the quaternary structure. We have discussed these interactions. Then let's see. State that globular proteins are generally soluble and have physiological role, and fibrous proteins are generally insoluble and have structural role. So there are two types of proteins. One are functional proteins, which have functional role, and the other are structural proteins. Structural proteins. Examples of functional proteins include enzymes, hormones, transport proteins, which is like hemoglobin, and we have this hemoglobin in our syllabus, so we'll discuss this in detail. Structural proteins are fibrous. These are globular. Functional proteins are globular. And water soluble. Whereas structural proteins are fibrous, fiber-like, and they have high tensile strength. An example of structural protein is collagen, which is in our syllabus. And other than collagen, we have keratin. So this is also fibrous. So you also know about the silk, which is again a protein, and that's also, a structural protein. So silk, keratin, and collagen are structural proteins. Okay, so let's see hemoglobin first. So this is I describe the structure of a molecule of hemoglobin as an example of globular protein, including the formation of its quaternary structure from two alpha chains and two beta chains and a heme group. So a hemoglobin molecule consists of two alpha globin. Alpha globin can be drawn, we can draw it this way. Okay, so this is coiled and folded and it makes a 3D shape. So this is how we can make, a 3D shape here. So this is alpha globin. Such two alpha globin are present. So I'll just copy it. Okay. So let's see. So we can, okay. So these are two alpha globin. This is also alpha globin. And such two beta globin are also present. Just a moment. Here. So we have this, okay. So this one is a beta globin. And we have another beta globin. So this is how four polypeptides are there in this hemoglobin molecule. Okay. Two alpha globin and two beta globin. These red are beta globin. We don't need to know why the names alpha globin and beta globin are given to these polypeptides. Now, each polypeptide, that has a 3D globular shape. So each has 3D globular shape. So it means that each polypeptide has tertiary structure. Then each polypeptide has, a heme group, a prosthetic group attached with them. So we can, okay, so this may be so we can draw, this is a prosthetic group. Prosthetic group mean a non-protein part attached with, each polypeptide. So we can draw this. This is a prosthetic group which is attached with each polypeptide, and it has an iron atom with each prosthetic group. Okay, so this is a prosthetic group. It has an Fe or iron atom there. So that's also known as a heme group. Okay. And this iron has, you know, an oxygen molecule can bind with this iron atom by cooperative bonding. So O2 binds with Fe with cooperative bonding. We don't need to know the details of cooperative dative bonding or cooperative bonding. Okay. So this binds with it. And consequently, as there are 4 Fe or atoms or iron atoms there in a hemoglobin molecule, so 4 oxygen molecules can attach with it. And we can write this as Hb. One hemoglobin molecule combines with 4 oxygen molecules, and that would give us an HbO8, which is an oxyhemoglobin. And this oxygen is transported in oxyhemoglobin form from the lungs to the other parts of the body, and this is the reversible reaction. Now, each polypeptide has hydrophilic R groups on its outer side. You know, this outside of these molecules has polar R groups on outwards. You know, we can write this R here, we can write R here, we can write here, an R here, an R here. We can write an R here, an R here. So these are hydrophilic or polar R groups. Polar R groups are outwards of this molecule, and they make hydrogen bonding with the water. Make hydrogen bonding with the water. This results in the hemoglobin molecule becoming a water soluble, and this also folds it into a 3D globular shape. So that is what is the quaternary structure, you know, as there is more than one polypeptide. So that's why it is called quaternary structure. Okay. There are also R groups directed inwards of this, you know, interior of this molecule. This has an R group here, this is an R group here, this is an R group here, this is an R group here. So these R groups are non-polar R groups. Okay. So non-polar R groups are towards the interior of this or inside of interior of the molecule. And this holds the 4 polypeptides together by hydrophobic interactions. So ये जो R groups हैं, these you know R groups, non-polar R groups hold the polypeptides together by hydrophobic interactions. Okay, so that's about hemoglobin. Let's see, we have a question on hemoglobin before we discuss collagen. We discuss some questions on hemoglobin. Figure 3.1 shows a molecule of hemoglobin. This is alpha globin and this is beta globin. Explain how a molecule of hemoglobin shows the 4 levels of organization of protein molecule. In this question, the question is not about this drawing and this not about this structure. This is generally a question about how hemoglobin structure shows these 4 levels. So primary structure, you know, number and order of amino acids or sequence of amino acids determine the primary level of a protein. So obviously hemoglobin has, you know, 4 polypeptides, 2 alpha globin and 2 beta globin. Alpha globin, each alpha globin has 141 amino acids, and beta globin has 146 amino acids. So you can multiply it with 2 and you can calculate the total number of amino acids in a hemoglobin molecule. And then if you look at the secondary structure, in secondary structure, hydrogen bonding between the amino acids in a polypeptide chain can coil a polypeptide into an alpha helix or into a beta pleated sheets. So all these three things are there in the secondary structure in a hemoglobin molecule. Tertiary structure, then each polypeptide has 3D shape, and each polypeptide has prosthetic group attached with it, so it indicates that this has a tertiary structure. And there are 4 polypeptides in hemoglobin, so that's why it has quaternary structure. So that's how you have to describe the each level of protein structure found in hemoglobin molecule. Okay. Now let's see. Yeah, that's the mark scheme here. Look at this primary structure. You already know primary structure is described by the sequence, arrangement, or order of amino acids, plus peptide linkages as well. That has not been given in this mark scheme, but that's true. Secondary structure by alpha helix or alpha helices, or description of alpha helices, or even the hydrogen bonding. Then tertiary structure, folding of each polypeptide globin, coiling, and held in place by interaction between the R groups. You can write about the different interactions, 3 or more named interactions. And the prosthetic group is already there. You see, this is heme group or prosthetic group. Quaternary structure, there are 4 polypeptides, 4 globin, 2 alpha and 2 beta globin, except chains instead of polypeptides or globin, and accept reference to more than one polypeptide if specific, to alpha and beta chains or alpha and beta globin. Okay, so that these are the same points that we describe or use them to describe the different levels of protein structure. Here we have hemoglobin is a globular protein which is able to transport oxygen and is soluble in water. It can transport oxygen and it is soluble in water. Explain how the structure of hemoglobin molecule makes it able to transport oxygen efficiently. So how it has an efficient transport of oxygen, you can say each polypeptide has a prosthetic group attached with it. So, and each prosthetic group has iron atom and one oxygen molecule binds with one iron atom. So 4 oxygen molecules can bind with a hemoglobin molecule by cooperative binding. Okay, so that's how you can answer this question. Now explain how the structure of hemoglobin molecule allows it to be soluble in water. So that's very simple. To make a hemoglobin molecule soluble in water, polar R groups are outwards of the hemoglobin molecule. These polar R groups make hydrogen bonding with water and make it water soluble. You can say these are hydrophilic, so make it soluble in water. There are just 2 points. So you can mention about the polar R groups or the hydrophilic R groups, and you can mention about their presence on the outer side of it, and which help them make hydrogen bonding with the water. Okay, so there are 2 points: polar R groups and hydrogen bonding. So let's see, this is another question on hemoglobin. Figure 3.1 is a diagram that highlights the tertiary and quaternary structure of a hemoglobin molecule. Explain how the tertiary and quaternary levels of protein structure of the hemoglobin molecule contribute to its role in transport of oxygen. So again, you know, the tertiary and the quaternary level, you can say about tertiary that it's, 3D globular, and it has, a heme group as a prosthetic group attached with it. And you can say there are 4 polypeptides in it. So there are 4 heme groups there. So 4 oxygen molecules combined with, the 4 prosthetic groups or 4 iron atoms present in this hemoglobin molecule. So that's how you can write about, explain how the tertiary and quaternary levels of protein structure of the hemoglobin molecule contribute to its role in the transport of oxygen. Let's see these points. You can say globular hai, this is rounded or globular. There are hydrophilic amino acids or R groups that face outwards. There are hydrophobic R groups that are toward the inside of it. This makes water soluble or soluble in the cytoplasm. And there is a heme group, prosthetic group, porphyrin ring, which binds with an oxygen. There are 4 polypeptides, so there are 4 oxygen molecules that can bind with it, and they bind by cooperative binding or the allosteric. Tertiary structure allows association of prosthetic groups. Okay, so this presence of prosthetic group with a polypeptide, that describes the tertiary structure. So that's how you can answer this question. We have another question here. As blood passes through the capillaries and the lungs, it becomes oxygenated. Okay. Explain how the structure of the hemoglobin aids the uptake of oxygen in the lungs. So this is again about hemoglobin. It's not about RBC. And it is for 4 marks. Again, you can say there are 4 polypeptides in hemoglobin. The hydrophilic R group pointing outwards, which makes soluble, which makes the hemoglobin molecule soluble in the water. And each polypeptide has 1 heme group. This heme group has an iron atom. Okay, and this makes temporary bonding of oxygen with each heme group. So consequently, 4 molecules of oxygen that bind with 4 prosthetic groups. This makes oxyhemoglobin, and this bonding is a cooperative binding, and that carries, the oxyhemoglobin that carries oxygen from lungs to the other parts of the body. So that's all about hemoglobin. Now we have collagen. Describe the structure. Describe the structure of a molecule of collagen as an example of fibrous protein and the arrangement of collagen molecule to form collagen fibers. So we look at the collagen molecule first, and then we will look at, the collagen fiber. Let's see a collagen molecule. Collagen molecule. A collagen molecule consists of 3 polypeptides that are each is a right-handed helix and tightly twisted around each other. So this is one polypeptide. We have another polypeptide, and we have another polypeptide. So there are three polypeptides that are twisted around each other, tightly twisted around each other. So a collagen molecule is a straight, unbranched molecule which has three polypeptides tightly twisted around each other. So if we look at one polypeptide, each polypeptide has a repeated sequence of, this is one polypeptide. Each polypeptide has a repeated sequence of 3 amino acids. Every third amino acid, if this is number 1 amino acid, this is number 2 amino acid, and this is number 3 amino acid. Every number 3 amino acid is, this is number 3, then we have 1, 2, and this is number 3. So every third amino acid is glycine. So a polypeptide has a repeated sequence of 3 amino acids. Okay, every third amino acid is glycine. So if we look at the structure of glycine, glycine is the smallest amino acid among all the 20 amino acids. It has the smallest R group and can be written as C, which has a central C, and this is carboxyl carbon, carboxyl group, and this is an amino group, and we have an H here. Here, instead of this R group, it has a hydrogen here. So this is the smallest R group that an amino acid can have, and this is called glycine. Okay, so this glycine has smallest R group. Presence of this glycine repeatedly in a polypeptide chain that contributes to the adaptation of this structure in the function of a collagen molecule in such a way that smallest R group allows polypeptide to bend slightly. This bending is not due to an alpha helix or hydrogen bonding. Instead, it is due to a smaller R group. And secondly, smaller R group allows, tight wounding or coiling of 3 polypeptides around each other. Okay, so if they are tightly wound around each other, it would provide high tensile strength to the collagen molecule. Now, such many collagen molecules are held parallel to each other, okay, in a collagen fiber. Okay, so if this is, for example, one collagen molecule which has 3 polypeptides twisted around each other, and another collagen molecule will be present here. This is another collagen molecule. You will see another collagen molecule here. Okay, you can see another collagen molecule here. This is another collagen molecule. Maybe there is another collagen molecule here. And another collagen molecule here. Okay, so you see, these are all collagen molecules that have 3 polypeptides twisted around each other. And these collagen molecules are held parallel to each other. in a collagen fiber. There are cross bridges formed in between, the ends of these, there are cross bridges between the ends of these molecules, which provides strength to a collagen fiber. Similarly, there are hydrogen bondings between, the adjacent collagen molecules that also increase the tensile strength. And one very important structural adaptation of collagen molecules and a collagen fiber which provide high tensile strength is this ending of these collagen molecule. You see, this collagen molecule has ending here, the other collagen molecule has an ending here, another collagen molecule has ending here. So this is an ending here. So do you see these all do not end up at the same point? This could have been in such a way where this is, for example, one collagen molecule, and this is another collagen molecule. This is, another collagen molecule. So if it had been like that, having these collagen molecules all ending at the same level in a collagen fiber, so this would leave the weak areas or weak lines at these points. So these would have been weak areas or weak lines along the length of the collagen fiber. As these are not ending at the same level, they are ending at the different levels. So these are known as ends of collagen molecules are stepped out of each other, or you can say that there are staggered endings. So this staggered ending or stepping out of these molecules from each other, that leaves no weak lines along the collagen fiber. So this provides higher tensile strength to the collagen fibers. Collagen is found in almost every part in our body. It is found in bones, it is found in skin, it is found in cartilage, nose bridge, ear, and almost everywhere in our body. And that provides high tensile strength to these tissues. And it is also found in, in the gums to hold the teeth in the gums. So that's all about the collagen today. We will discuss some questions on collagen, and we will further understand the role or the adaptation of structure of collagen molecule in its function. जी.
तो ये जो पॉलीपेप्टाइड चेन्स होते हैं कोलेजन मॉलिक्यूल के अंदर, ये कोवलेंट बॉन्डिंग और हाइड्रोजन बॉन्डिंग के थ्रू जॉइन होती है ना?
Yes, ये जो क्रॉस ब्रिजेस हैं here, these are the covalent bonding. Okay. So इनके जो कार्बन टर्मिनल और नाइट्रोजन टर्मिनल ऑफ दीज पॉलीपेप्टाइड चेन्स in the adjacent जो molecules हैं that make the cross bridges or that make the covalent bonding in between them. Is it okay?
Yes.
So that's it for now. See you tomorrow, inshallah.
Allah
Allah Hafiz.